today we're going to talk about single-cell RNA-seq and single-cell ATAC-seq data analysis and integration. I will talk about how to import the data, QC the data. We're going to briefly talk about the single-cell RNA-seq data analysis pipeline. For more details, you can refer to the single-cell RNA-seq data analysis videos. And we're going to more focus on single-cell ATAC-seq data analysis pipeline, that how to build that in Partek Flow. And at the very end, I will show you options that how you can integrate those two assays. All right. Okay, so the data set I'm going to use is downloaded from 10x website. If you go to their website, the data set, you can find all kinds of assays. So the data I'm using today is a multi-ohm ATAC and the gene expression data set. We're going to start from cell ranger output data. With those two assays together, the output file is kind of, there's multiple files, kind of more complicated than what we learned if you just import a single cell RNA-seq data. So here, in this page, I just want to mention that this is the cell ranger output file folder they generated. And if you click on the format details, you will know, those are the file formats. And for Flow, when we import the data, we need 5 files. So here, let's see, maybe I should go back to this one. So there is one file called the per barcode summary matrix. It's .csv. When you download that, sometimes they call it like a single cell .csv. Let me just show you, the 5 files, what this means. And sometimes they call it a per barcode matrix .csv. I guess it depends on what version of the software you're using, the cell ranger ARC you're using. They might have different names, but anyway, it's .csv file. And the file format will look like this. So within this file, they have the barcode for, for both assays, and then I think this is the final barcode. But within this, they have some summary information about how cell ranger QC the data, give you some summary information, how many reads or how many fragments, those kind of information. So we use this file to import. That way, when we do some QCs, we don't need to recompute the information. Okay, so this is one file we need. I'll just show you the format of the file for each one. This one is a filtered feature barcode matrix. This is very similar to single cell RNA-seq. We're familiar with this. It's a binary format, and there is a text format. It's a directory filtered feature barcode. Those two containing the same information. We want you to use the filtered one, not the raw one. So the format is the same, but this is the raw. They have a lot of empty barcode there. So always use the filtered one. You can choose either one. We support either one, but H5 file is much better. It's much smaller, and it's only one file per sample. It's easy. This one contains 3 files. Let me show you what they look like. So this is the H5 file. And then if you download the folder, unzip it, this is going to be a folder with the 3 files. And you probably already familiar with that. We have a barcode information, we have a feature information, and then this is the sparse matrix. Okay. All this containing, if you only have ATAC-seq, this will containing only the peaks. If you have, RNA-seq, you only have the genes, but here, the barcode containing both genes and peaks together. So it's much bigger in terms of number of features. And then there is another one is called fragment TSV file. So this is the real file, and this is the index. Index, you can see the file is much, much bigger. With the index, it will be much quicker to access the file. Let me show you what this looks like. And this is the fragment.tsv. Okay. If I open that. So in the fragment.tsv, view containing, it's kind of very much like a bed file. They have a chromosome start and stop for each fragment in each barcode, which means in each cell. So for every cell, they have the fragment, and the fragment you can have multiple reads, and this is the number of reads. But you will see that different cells, the start and stop might be different. You know, it's almost like for each cell is a peak, but it's not. You know, they're very low. Very low coverage for each cell, and also different cells, they are not going to land up. So this is one of the files. We sometimes need to exit this file. So typically, only when you have multiple samples, we need this file to combine all the different samples together, because you have to make all the samples for all the cells, the start and stop for each peaks, they lined up. Otherwise, you cannot make a matrix. So for single cell, for single sample, actually, even we require for you to import this, but we don't really use that. What we need is, and there's another file called peaks.bed file. And this file format is very much the same as the other one, you know, but this is lined up for all the cells, and you only have a chromosome start and stop location. So all cells, they will have the same information, the same location. You know, you think about it for every gene, you know, every gene, they line up for every sample, for every cell, right? So actually, this file for the peaks, for the ATAC-seq is exactly the same as if you only have ATAC-seq data, you don't have a gene expression, you're going to have a peaks.tsv.gz. So it's included in this, you know, filtered barcode matrix folder. But you don't need to worry about it, how they similar. I just want to let you know, because this is your feature location, or how you define each feature, how you define each peak in ATAC-seq. So this is, as you can see, it's kind of more complicated for this type of assay. Okay, there's yeah, so that's all those 5 files, 1, 2, 3. Each 5 file is 4, and then the single cell or per barcode matrix.csv, so 5 files. Let me show you again what we need here. Basically, just ignore all this. You need to fragment TSV, fragment index. So those two, they're together. Peaks.bed file, the count matrix, H5 format, and then per barcode matrix.csv. So those are 5 files. There's an extra file called the BDPE files, the featured linkage. This file is a secondary analysis for cell ranger. They can do secondary analysis, and which they will have a UMAP, you know, TSNE, those kind of information. I will show you what this folder will look like. If you download that as a zip file with the secondary analysis, you know, cell ranger do some initial clustering. They also have a file called feature linkage. If you do RNA-seq and ATAC-seq together, they will have this, and we need this file to display the linkage between RNA-seq and the ATAC6. This is the one way of integration. So if you want to visualize that information, we need this file. Okay. All right. So that's all the files we need from 10x website. I'm going to close those. And also, if you need more details, you just click on the file format details, and that will take you to this page. You can check. There's definition on what does a file format look like. You know, the peaks, they have information about that. So it's on this page. Close that. All right, so this is what the pipeline would look like. I know it's kind of scary, but once I show you what I did, it will not be that bad. First of all, I'm going to show you how to import the data. We'll click. I'll just give a name of this project. You're going to add the data. And we'll choose using single cell ATAC. Even you have a single cell ATAC or a single cell RNA, we're going to use it. As long as you have the ATAC data, we're going to use the ATAC importer, okay? Because the only information for RNA is in the H5 file. So the H5 file has the peak count information and the gene count information for every cell. We will import the cell ranger output, and also here again tell you which 5 files we need. Okay. Now we're going to add a sample. Again, we tell you all those file formats. You're going to browse to the files. Select sample. I want to select all those 5 files. Okay. Fragment with the index, batch file, count matrix H5, and the per barcode CSV file. Okay. You're going to give a name. So suppose it's my, this is a 3K PBMC human sample, just give a meaningful name. If you have another sample, do the same thing. Just add a sample and browse the file. The feature annotation for peaks, you don't need annotation because the peak name containing the annotation information, the location, and the annotation is mainly for single cell RNA-seq data. So you're going to specify what your gene annotation coming from. This is a human data, so your upstream will tell you, okay, which ensemble version you're using. This is exactly the same as a single cell RNA-seq. And you can choose. All the bottom is pretty much the same, and then you're going to click finish. All right, so now I'm going to switch to my pre-built pipeline, and I will show you the steps. So pretend you don't see anything. Right now, I only have this one sample. Okay, one sample with almost 3,000 cells in it. And you can see the number of features is very big. It really depends on your read depth, how many peaks you detect. It's not like a gene expression, you always have like a 30,000 genes. And this can vary a lot, and the peaks can have a lot. We have a 134,000 features. Actually, only 30,000 features per gene. The rest of hundreds, thousands of features, they are ATAC. Okay. And now you can do what we learned before, do pre-alignment QA/QC. Is the same. Actually, the concept is very much the same. So once you perform single cell QA/QC, you're going to have the QC on the ATAC data, and you have the QC on the RNA. RNA, we're kind of familiar. We have the look at the y-axis. This is the total number of UMI count. This is the total number of the genes we detected. They are non-zero mitochondrial percentage, ribosome percentage, right? For ATAC, we have a similar situation. This is the read depths, the total number of count in for each cell. and total number of the peaks, regions we detect in each cell, so, which is kind of corresponding to number of genes that is non-zero. I can make them lined up a little better. Okay. But this one is a new one, and this is called kind of a signal to the nucleosome signal ratio. It's a ratio of mononucleosome versus nucleosome-free region. How they calculated that is the mononucleosome, they just look at the fragment. So the fragment, so we saw that file, look at the length. If the length is between 147 to 290-ish, in that length, they will call that, they think that is a mononucleosome. The nucleosome-free regions usually is less than 147. So they just calculate the number of the fragment, get the ratio. And some paper mentioned that the ratio shouldn't be more than 4. But as you do the QC, when you do the filter, you should always look at your distribution. I can see they are less than around I would say 0.6 or 0.7 in this area. Okay, so always use your data to perform the filter. The TSS enrichment score is they calculate it's a kind of a signal versus noise ratio. Basically, they calculate the coverage at the TSS location versus some using the flanking regions average read depths. They will call that as noise. They kind of go up and down. I think it's a 2,000 base pair. They calculate the end of this TSS flanking regions and use that as a background and to calculate the ratio. And here, the higher the better. Low, that means you don't have enrichment in your TSS region, so you want the higher the better. But still, if it's too high, that could be a doublet. But you want this to look at your data to determine your filter. For gene expression, and we all know how to perform this filter. And you go to filter and select and filter and adjust based on the histogram and choose your threshold to select the cells and to select what you think is good, valid, viable cells. And then after you select, you perform filter applied to your data. Okay, it's the same concept. And as for the cutoff, always look at your data. All right, so after you filter your data, so I did clean up a little bit, and then you're going to perform filter features. Again, we're going to kind of use a very permissive filter. So for every cell, you will see the filter feature dialog. We're going to, in the next week, you're going to see the new release. The dialog kind of changed. But we have more information here. It's the same thing, but just it's changed to different options. Before we list the options in one page, it's kind of too long, but now we choose to do it this way. So you use noise reduction, and you can choose, if the value basically now it's a raw count value, right? If the raw count value is 0, you don't have a less than, and in what percentage of your data. If you only have a 2,000 cells, less than 3,000 cells, and you say, hey, 99% or 99.5% of the cells, I will filter that out. Okay, so that will help you reduce your data. Your downstream analysis will be faster and also reduce the noise. And then when you mouse over, before I got 134,000 features, and here I got 95,000 features, which including both genes and peaks. I use the same criteria to filter. But you can do this separately. For genes or peaks, you can do that separately. But I would recommend you, when you filter cells, do it all together. okay? Because later on, you don't want some cells, you only have ATAC information, but not gene expression information, or vice versa. I want all the cells I want to keep, they have both the gene and the ATAC information. Okay, so after I clean it up, the next step, if you have two assays, we will choose split by feature type, because those two assays cell ranger detect, one is gene expression, one is called a peak. So if you just click on this button, that will automatically separate the features. And now if I mouse over on this one, actually for gene expression, we filtered quite a bit. There's only 13,000 genes, 13,000 features. For peaks, there are 82,000 peaks. Now from here, you can, I think you should analyze them separately, because one's DNA, one's RNA, they are kind of a different perspective. I think you should analyze them separately. But sometimes if you want to visualize them together, that's fine. You might ask different questions. So for the single cell RNA-seq analysis, you will be familiar with, and we normalize the data, and then we're going to do dimension reduction, do PCA, UMAP, or do some clustering analysis, generate biomarkers to help us identify your cell type. I heard a lot of biologists told me that using gene expression to do cell type classification is better than ATAC. Well, single cell, the biggest job is cell type classification, right? So for gene expression, they said it is cleaner and the signal is better versus noise. Also, if you look for any biomarkers or any database, all the databases are based on the gene expression. They will give you the name of the gene. They won't give you the location or the chromatin opening regions as a marker. So for that reason, I think you probably can use the gene expression data to do cell type classification. And the method is exactly the same. You can go to our single cell RNA-seq data analysis, those two videos to look at it for details. But I just want to mention that when I perform all that, I generate a UMAP. I use UMAP based on the graph-based cluster information, based on gene expression, identify the cell type. And once the cell type identified, I can perform differential analysis and generate genes. There's a difference between cell type or between phenotype, all the different comparison. If you have multiple samples, you can do so. Okay, so this part I think we're familiar with. Now let's take a look at the peaks. For the peaks, we only have the location, a chromosome start and stop. You definitely want to know, okay, is this location, how they related to a gene? Is it in the gene's exonic region, intronic region, or intergenic region, right? Or promoter region. We want to annotate the peaks. So that's the first step I would like to do. As you can see, I use a different color to indicate the different kind of assays. I want to use the yellow represent the peaks. So now we're going to go to peak analysis, do annotate region. This is very much the same as when you do bulk ATAC-seq. We're going to utilize some single cell technique we learned from single cell RNA-seq, and also some ATAC-seq kind of a concept from our bulk ATAC-seq. So annotate regions, you're going to specify a gene annotation file. And also here tell you, do you want to report one gene per region? If you do one gene per region, this is what we're going to report, or that's the precedency, because your region might overlap with the multiple transcript, I should say transcript, not one gene, right? With the multiple transcript, maybe it's in one transcript intronic region, but the other transcript exonic region, right? If that is the case, we're going to report TSS first, and then TTS, and then CDS exon, and then UTR exon, 5 prime, 3 prime, and then, you know, if none of the regions not overlapping with any genes of those regions, we report as another gene's intronic region. If nothing than that, this is the report intergenic region. So that's the order we're going to report. Or if you choose this, we'll give you all the genes with all the regions. Depends on what you want to do with the table. For the downstream analysis in Flow, I think this one is cleaner. This one, if you really want to get to focus on a specific region, you want to see everything, I think this is better. But you can see it's going to be kind of messy. What if they overlap with like a 20 different transcript? It's kind of messy there. Also, you need to just to specify kind of your promoter region, right? So there's no definition of the promoter region in the gene annotation file. You kind of try to say, okay, usually people will see in the TSS upstream certain number of base pair or downstream certain number of base pair around the TSS we think is a promoter region. By default, we use 1,000, but you can change this. And also we allowed you to define the TTS region, the transcription termination site. You can, you know, give a number here and then click finish. So now this information will carry on to the downstream. We know the regions, and later on when you do any analysis, we can always pull gene information here. Now, here we have another two options to do. One is, suppose you really focus on the promoter region. I don't care about all the other things. You know, I don't care about all the peaks that happened in intergenic region or intronic region. I only cared about the TSS region. So we can just sum up all the promoter regions, the reads in there. So this step is you will do a promoter sum matrix. So we will create a matrix. This matrix is, you imagine each row is a cell and each column is going to be a promoter of a transcript. But here, this is where using Cell Ranger's algorithm, they kind of combined all the transcript at the gene level. So they have a definite, you know, they just combine all the transcript using one gene as a unit to represent the promoter sum. You sum up all the reads in the promoter region for every gene. So the result you will see this is going to be a similar number of a gene number. Right? So before we have 82,000 features. Once you generate this matrix, this is 14,000 features. Actually, it's a gene. We use the gene, but this gene is not a gene expression. Is the gene? We just use the gene name, to help you identify. You look at the promoter of the genes, the read count. So the downstream analysis is very much the same. You can, think this is a gene, but the measurement is only the promoter region. And from that, we can use a similar, single-cell RNA-seq pipeline. We perform PCA and a UMAP. So I get another UMAP. I call that the promoter UMAP. And also you can perform differential analysis between, once your cell type identified here, even I call this as a gene, but you will remember this is actually a promoter region. The location, we call that is the gene location, but you remember this is a promoter region difference. Okay. This is one way to look at the data. The other way is just to look at the peak. I just want to analyze the peak coverage. I don't want to, summarize to any aggregate all the peaks in any other format. But I annotate the peaks. I know the peak location. We might have peaks in intronic region, etc. Here, I want to introduce you another normalization method. For RNA-seq, we just use log CPM, but for ATAC-seq, this is in this industry. We kind of follow the scNAC pipeline, the SARA scNAC pipeline, and they recommended for ATAC-seq the normalization. They are using IDF normalization. If you mouse over, you can see the full name. It's a term frequency, inverse document frequency. To be honest, I don't understand what that means. This is in the scNAC R package. We just wrap it up. So the whole idea is they kind of normalize the data both ways, both at the cell level, try to correct the differences in the cell depths, and also across all the peaks. And they give higher values to more rare peaks. But the bottom line is you will know after this normalization, the data is in a log E space. So this is after normalization, you can download it. So click on any circles, you can download the data, but it's going to be huge. But the value is going to be in the log E space. Okay. So this is a different type of normalization. It's kind of in this field, everybody is using normalization. If you want to try, you can try different other normalizations too, right? Also, we're gonna, after normalize the data, we're gonna do the same strategy, gonna detect, visualize the data, perform UMAP. And before we always use the PCA, and here we're using a different dimension reduction method. It's called a singular value decomposition. So if you go to exploratory analysis, you will have this SVD. It's a singular value decomposition method. Again, as you can see, we use the scNAC package because this package is very popular for ATAC-seq data analysis. So we just follow that pipeline. To be honest, the result is kind of similar to PCA. I would say PCA is one type of SVD, or they are all dimension reduction function, but SVD is kind of utilized for all other fields rather than the genomic field. So just a dimension reduction method. You can, of course, use PCA to do that too. And then after this dimension reduction, you can treat it as PCA. You can perform harmony on this guy. You can perform UMAP, graph-based clustering on this guy too. So I performed the UMAP on this. So you see that we have a UMAP of a gene expression. Always right-click and rename that. You know what this is. And I use this to this green color represent the promoter information. I have a UMAP for promoter. Also, I have a UMAP for peaks for ATAC-seq. Another thing is when we learn the site-seq, you can visualize the different assay by combining them together. And the method we used is WNN, or where's my WNN? So multi-model neighbors. So this task requires you to click combine PCA data node or SVD data node. So if I choose ATAC SVD data node, and if you go to find the multimodal neighbors, you need to choose another one. You can combine with the gene expression PCA or promoter PCA, or yeah, that's the only two options. So you choose this guy, and then click finish. So SVD and the PCA, they can combine together. And this branch is what I combined ATAC and the gene expression together. And then I will use UMAP. So there's another UMAP. I call that WNN UMAP. Weighted nearest neighbor. So now let's take a look at. Oh, sorry. I probably didn't open that. So this is my for you map. I already saved it. And this data, based on my gene expression, I already classified that in, this different cell type. We can, color all that. Now you can compare side by side. We can clearly see, for example, the B cells, no matter what you map, they all look very separate, right? From all the different angle, they are all kind of separate. So the B cell is separated very well. But there are some cell types, probably one SE captured better than the other. For example, like this CD16 monocyte, and this is general monocyte. I couldn't classify anymore based on my limited knowledge of biology. For the gene expression, we can see there is a little bit of separation over here, but if you use the ATAC, you cannot see that, right? You won't be able to see. If I don't color that, you probably think they are the same. But if you use the WNN, also, they are very kind of close, but it's not separated very well. And this promoter is going to be really noisy here. And also there are some subclusters we can see in ATAC rather than gene expression. For example, over here, this is the T cell. I didn't put the legend here. So this color is the T cell. My gene expression T cell is over here. If I only look at gene expression, I see, okay, they clustered together. Maybe I see there's a separate, but I'm not very sure, right? But for ATAC, I see a two separate cluster here. If I selected, if I look at gene expression, oh, they kind of on one side of a gene expression, but the ATAC separate better. So some features, one SC might see that better than the other. So I'm not sure what this is. If you don't know what that is, maybe what we can do is, my limited knowledge, I only know T cells, CD4, CD8. We can take a look at that. So suppose I use the gene expression data, because usually biomarkers is using gene, right? I search for CD4. CD4 gene. I can drag this gene to color that. I'm gonna put in the green color. I can see, there. I don't worry about all the other, maybe NK cells, they have a CD4 high expression. But this is the T cell location, right? And then if I go to CD8, CD8A, oh, wait, hold on. I forgot what my thing is. I'm gonna duplicate that and I use this as cell type. Oh, this is T cell, sorry. So for gene expression, we can see here, this is the T cell, and those part have a high expression of CD4. In the middle, they have a high expression of CD8. Well, you can, you might have to use other evidence to see those are different population, right? And in the meantime, if I use the CD4 to color this guy, and the CD8 to color ATAC, oh, sorry. I'm going to use a CD4 and put on and I can see here we have more CD8, right? And over here they have more CD4. But of course, when you see some conflict information, you might need more evidence to identify the subpopulation. Okay, so that's why multimodal will give you more perspective to look at the data. And I know some people even put cell surface protein on this kind of assay that will, like a SciSeq, it will provide another layer of the information. Okay, so that's how you utilize multimodal for visualizations to help you more accurately identify the cell type. All right. So that's the okay. Now I want to talk about once the cell type is identified using, all those math techniques we learned before. And then besides we perform differential analysis on the gene expression, we also can perform differential analysis at the peaks and also at the promoter regions. So this is the peak information, differential analysis. 82,000 features. And this is the peak ID. And the peak ID, they have a chromosome, colon, start, and stop. And also because we annotate that, we know which genes, which transcript, especially when we annotate that, we kind of use the transcript information. And this is the section talking about, this region overlap with how it is related to the gene, and gene name, transcript name. After that is the expression. Sometimes if the table is too big, you go to optional column, you can turn off something that you don't care. So make it smaller, or you can turn off, I think the gene name, gene section is the important. ID, it's just a number, it's not that important. You can add, also you can add like a % of the overlap. What is the % of the region overlap with the transcript and the % of the peak overlap to the peaks. When you see this is 1, this is a fraction, that means the gene is much bigger than your peak. So this peak is completely within the gene. When you see, the other way, your peak, it will be bigger than the transcript, or sometimes they can be partially, like this one, they just partially overlap. Only half of the peaks is within this gene. Only a tiny bit of the region of the gene is overlap the peak. So you know what exactly the location is. And the rest of them is the same as any differential analysis report, the p-value, fold change, mean, etc. So on the left side, we can perform filter. Of course, you can look at volcano plot. It's just the same as we learned before. Okay, you just have more dots because there are more peaks than genes. Okay. And then on the left side, we're going to use, suppose, FDR 0.001, use your cutoff. And with the fold change, suppose I want to use, 2-fold change. Also, you can add some other filter information. If you say, hey, I really only want to focus on TSS, and you can go to the gene section, say, hey, only give me TSS region. So now I only have this, 2,000 regions you can create to generate a filtered data node. Okay. Another thing is I want to highlight that whenever you have a differential analysis, you can always perform filter task. That filter is on the report itself, but the filter task will give you more flexibility. So if you choose a differential analysis filter, you can filter based on metadata. So basically the metadata means I want to filter based on, suppose I only want to look at if they overlap with the gene, I wanted the gene, the biotype has to be like a protein coding or something like that, right? And I want, you can specify p-value, all the statistic there, and or those kind of information. So this one is more flexible for you to do and or information and or operation, but the other one on the the report filter, you only do AND operation. Okay, so I just want to tell you, you can perform the filter to generate a significant list. So here, I generate a significant list of genes. I compare B cell versus T cells. Since I only have one sample, I only compare the cell type. This is significant genes list, and this is a significant promoter list. And here is a significant region list. Okay, and you can compare them. One way to compare them is using Venn diagram, because they all have a gene information. You have to make sure, what genes you want to look at in the region. Suppose I don't want to look at if they overlap with a gene in the intronic region, I don't want to look at that, then you should filter out intronic region. Okay, but make sure you cared about all those regions, and you can compare using gene name as a bridge to highlight, link them together. So I will choose the gene name here. So suppose this data node, make that informative. This is a gene expression, and this green branch, this is my promoter region. I'll call that promoter. And the promoter, the initial ID is the peak. Actually, it's not really peak. It's the peak around this gene. You can use that, but to make them consistent, I just use the gene name. The other one is the significant region. So this is my ATAC, right? ATAC, the peaks, or you can call that peaks. And this one is the region, they won't overlap. I need to select my gene name. And now you go to display selection. That's the Venn diagram. Okay. To be honest, I don't know all the biology, how to interpret that, all the comments, maybe those genes, you really should focus on, because this is all between T cell versus B cells. And if the promoter is significantly different, maybe the promoter will regulate the gene, so you can interpret the biology here. And once you have a gene list, you can choose this gene list to download it, to save it in your list management, or you can perform a filter on it. Say, hey, I want to filter based on whatever data node. So you can pick a data node to filter on it. So this is one way of how to integrate different assays by using a gene name as a bridge to link different assays together. Another way is you can, just like when we do bulk RNA-seq and ATAC-seq analysis, we can, if every cells you perform 2 assays, we can perform a correlation. However, for single cell, because there's a lot of zeros, your correlation won't be good. So that's just the nature of the single cell in the current technology. So to perform that, you have to perform on the count matrix data node. So I will choose normalized count matrix data node. I don't want to look at all the 13,000 genes to correlate with the here, this is normalized, to correlate with 80,000 features. So that computation is going to be very expensive, and the table is going to be huge, right? So I would recommend you only use your significant one. So you're going to use your significant regions to filter your normalized count. So basically, once you have a feature list, you save it, and then when you choose a filter, you're going to use the filter feature and choose a filter based on a list. So if you already create a list and you choose the list and filter that, and then this is how I performed before, 2248 cells with 82,000 features. After filter, I only focus on 2248 cells, still the same, but only 600 features. Okay, so that's significant ones, and do the same thing for RNA. So Once you have a count matrix, of course, you want to use normalized count. Click on this count matrix. You go to statistics and perform a correlation. You're going to choose to correlate across assays. This is exactly the same method we use for bulk RNA-seq and ATAC-seq. You're going to choose another data node, and this is the one. When you perform the correlation, one thing is since all the genes and regions, the peak regions, they have a genomic location, you might only want to correlate the genes or the regions with the genes on the same chromosome, right? So maybe it's an enhancer, they can be in the far distance if that's the case, or you can choose all the combination. You might have gene on chromosome 1, the reason on chromosome X, you can perform correlation. I don't know how you interpret the result. If they have a high correlation, does that mean that they are related, or yeah? So I think most people only need on the same chromosome. For single cell, because there's a lot of zeros, I think using rank Spearman correlation, which is a rank correlation, might be better. But still, it won't be very good. And also, you can choose your p-value and using the correlation coefficient, the absolute value as a certain cutoff to report it. I tried to use this, but they kind of all filtered it out. So then nobody will pass. I think the highest one is 0.5 something. So I end up, I just want to show you this function. This is the correlation coefficient list. It's just because all the zeros there. You can see the highest coefficients of 0.57, which means actually it's no correlation. Nobody will look at 0.5. And this, again, is the name of the regions, and then which genes relate to this region, which transcript relate to this region. And the feature 2 is my gene expression data and the chromosome information here. If you click on this correlation plot, it's a 2D scatter plot, and you see, it's not that great because most of them, they're gonna have zeros. Either they have a 0 in ATAC or they have zeros in gene expression, and this one is both are 0. There's only a few number of cells that they have a high expression. Of course, you can perform, but you have to do it for different correlations separately, just to filter down only to the non-zero for each SE and then perform a correlation. Okay, so that is the how you integrate with the 2 SEs. Another thing is just for visualization purpose, we just take the cell ranger output result. Currently, Partek's chromosome view is not available to visualize the count matrix yet. We are going to add that. Our chromosome view mainly is only for BAM file, but when we import cell ranger output, we don't need BAM file, but we're gonna add more visualization in our chromosome view. But in the meantime, you can use cell ranger's output, the linkage analysis, to visualize how the linkage is going on. To do that, again, we need to go to their secondary analysis output folder, and there is a linkage file. It's called BAD-PE file. So that's the file we need. You click on your single cell data and you go to peak analysis, and there's a feature linkage analysis, and the Partek will ask you to specify that file. So you just browse to that file you uploaded, and then it will generate. Actually, we embedded IGV in Partek Flow, so we'll give you this data node. It's purely for visualization, and if you click on that, that will give you this. If you're familiar with the IGV, and this is what IGV is. IGV is a desktop application. We just wrap it up within this browser. I'm not very familiar with the IGV. You might be more familiar with it, so you can focus on certain chromosome. They have different tracks. All the genome browsers are kind of the same. They have different tracks, and then this is the linkage, and you can zoom in, to certain location, and they have the gene annotation and the peaks. At the bottom is the cell ranger calculated the linkage between gene expression and the ATAC information, and they give you the distance. I think this is nice, and we are going to add more features for in Flow for this kind of integration, but currently, to be honest, they perform each sample separately. If you have multiple samples, that's going to have some limitation to perform this type of analysis. You're going to have a lot. Only I think it's useful that if you know what a particular gene you cared about or on your significant list, that, and maybe you can focus on that particular gene and to see, okay, which regions relate to the gene, or a particular region and which genes, correlated with this regions. That means this gene will regulate, this region will regulate the gene, right? So that's another part. Oh, one more thing I forgot to mention, that for the ATAC-seq result, of course, you can perform, pathway analysis because you have a gene name. You can perform enrichment analysis, and for ATAC specific, you will do motif detection. This is exactly the same as bulk ATAC-seq analysis. You can detect motif, specify how many you want to look at. I would recommend you should look at one at a time. So hopefully, your enriched is for only one type of the protein you're looking for. And then the results will give you the sequence logo, give you the consensus sequence, and the details we detected in which location in your data. And the other one is you search for known motifs. We use the JASPAR database, and you click finish. That will give you the result. Small p-value is better, and you can click on, this is the detail where we found it, for which motif, and for a particular motif, you can go to the JASPAR database to get more information.